Hi, I'm Tom Shea. I'm a professor of biological sciences at the University of Massachusetts, and I direct the Center for Cellular Neurobiology. I'm also now the science advisor for SIVO Nutraceuticals. And today I'll be talking to you about keeping your brain healthy and your memory sharp. I'll summarize my academic team's laboratory studies, which spanned 20+ years and resulted in a formulation that optimizes brain function. Using that formulation, we conducted 7 clinical trials, starting in 2006 and finishing last year. And there's an entire host of people that made up the research and academic team that conducted these trials. Some nurses who also had PhDs, some MDs, some very dedicated graduate students who actually earned their PhD doing this work. A little bit about me. I was an associate professor at Harvard Medical School, and in 1994 I had the good fortune to relocate to UMass Lowell. The wonderful part of UMass Lowell is there is a clinical nutritional department, there are nursing departments. So working with those people, the work we did morphed from a basic neuroscience work into nutritional neuroscience, and that was where the formulation arose from. Some of my kudos, if you will. I have over 300 scientific publications. I've gathered quite a bit of money in research funding over the years. I've had the fortune of having many doctoral students in my laboratory, many master students, and I present seminars and workshops regularly for the Alzheimer's Association and for regional organizations such as senior centers. Well, let's talk a little bit about the research itself and brain health. Now, everything about us gets slower as we age, and that includes our brain. Our brain isn't a muscle, but you have to think of it as one. You have to exercise your muscles to keep them strong. You also have to exercise your brain to keep it sharp. As we age, remembering things becomes more and more difficult. This becomes even more difficult if you suffer from dementia. But dementia or not, we are all going to face an age-related slowing and a challenge to our memory. If you do suffer from dementia, there are many prescription drugs that can help. Unfortunately, you have to wait till you decline considerably before you warrant a prescription. What do I mean? You have to decline to a point that your family and your doctor recognizes that you've declined. This means it's late in the game to apply the prescription. Ideally, you want to take action before your brain declines. Now, that's where we focused our research efforts. Here's a little diagram of brain and brain cells. Now, up in the corner, there's a side view of the brain. And a brain cell, scientifically referred to as a neuron, is a funny little cell. It has many inputs, and in the center of it, it has to gather together all those inputs and make a singular output. What do I mean by that? Well, you can see 1, 2, 3, 4, 5, 6 different inputs. Imagine 3 of them say yes, fire a signal, and 2 of them say don't. Well, that's a net of 1+, so the brain cell will fire a signal and pass it on to the next cell. At other times, there are no signals coming forward. At other times, there could be 3 or 4 inhibitory signals and only 2 signals saying fire, so the brain cell will not fire. All of these cells working together, firing or not, is how we think, how we recall our memories, how we form new memories. And a close-up down on the bottom shows brain cell number 1 connecting with brain cell number 2. I like to think of this like the Kung Fu salute. The first brain cell comes down onto the second one. And they're not directly connected. There's a little space in between them. What brain cell number 1 has to do is release neurotransmitters. And releasing those neurotransmitters tells brain cell number 2 to fire or not fire. And this goes on and on throughout our brain. Now, if you think about it, as we age, our neurotransmitters decline. You can reach a point where you have 2 perfectly healthy and active brain cells, but they can't communicate because there are too few neurotransmitters. So that's one of the reasons that brain activity becomes more challenging as we age. Another issue is we lose brain cells as we age. Now, this little diagram is showing you we have a population of brain cells as we age that is old, but it's healthy. I'm 61 years old. I have brain cells that are 61 years old, but they're still working well. I've got another population that's colored in yellow. It's damaged, it's tired, it's undergone the ravages of my 61 years of life. But you know what? If I feed it well and take good care of it, and if I keep my brain active, I can kind of scoot it back up to the green population. However, I also have a population that's a little too damaged, especially if I haven't been treating myself as well as I could, and it's too late to bring them back. So the challenge is to keep them, they're going to get older, but the challenge is to keep them healthy. Why do we lose brain cells? We simply live too long. Now, that doesn't mean I don't want to live until I'm 500 years old. I do. But we are biological creatures, rather fragile at that. And we're subject to all the problems of the earth that every biological creature experiences. And you may not think about it, but let's go back in time. We used to live to about 25 or 30 years old. Now we're living much longer than that. Well, you know, most animals live until they can have their babies, and then they live that number of years again. And they do something very beautiful. They die, and they make room for new animals. Well, we've changed that, haven't we? We can have our children at 15, 16, 17 years of age. We don't because of society. We've changed how we live. But that means 15, 16, or 17, pretty much our allotted time on the planet was about 30 years old, 31, 32, and that's how long we used to live. And how long do we live now? Well, 25, 30 years, another 25 or 30, and another 25 or 30. We're pushing 100 years, and as time goes on, we'll live even longer. So we're stretching out our lifetime longer and longer and longer. Now, there's a problem with that. Your skin is replaced all the time. Your liver is replaced all the time. Your intestinal cells are replaced. Your blood is replaced. The part of you that isn't replaced is your brain. You're born with all the brain cells you get, and you don't get any more. So as we live longer and longer and longer, it's like trying to get a car to drive 1,000,000 miles without changing the engine. You can fix the brakes, you can fix the transmission, the engine wears out. Our brain wears out. It simply gets tired, and it's not able to last. My brain cells are 61 years old. Come on, that's a very long time. And really, it was only to be about 25 or 30 years originally. So I've doubled my time, and I still want more time. Now, you've probably heard of stem cells. There are stem cells in all the tissues of our body. So it is possible for us to replace a few brain cells. However, we're not able to do the kind of replacement we want. You can tell already. Why? Well, there wouldn't be any Parkinson's disease. There wouldn't be any Alzheimer's. There wouldn't be any age-related decline. Someday, stem cells will be able to do this. We simply can't do it yet. Now, we're losing brain cells. All of us are losing them. People that suffer from dementia lose brain cells even more quickly. And let's think about dementia for a minute. Alzheimer's disease. Dementia is a broad category. It's almost like saying fruit. And within fruit, you can have apples, cherries, oranges, and so on. Alzheimer's disease is one form of dementia. There are others. But did you know that 10% of the people in the United States, age 65 or greater, have diagnosed Alzheimer's disease? 20% of the people, age 75 or greater, have Alzheimer's disease. It doubled in 10 years. 40% of the people aged 85 or greater have Alzheimer's disease, and a whopping 80% of those of us 95 or greater have Alzheimer's disease. Get out your calculators. What's the next iteration? What percent and what year? At 105 years of age, we will all have Alzheimer's disease. Now, that's an average. There are certainly people that do not. You may have that relative, Uncle Milt, and he's 110, he's sharp as a tack. Uncle Milt's on a long timeline. Some of us are on a shorter one. But the point I'm trying to make is, on average, we will all experience age-related dementia, or age-related heart disease, or certain cancers that come with age. This is the downside of living longer and longer. And as we've cured or conquered diseases like polio and tuberculosis, more of us live into this long-term effect. Where do we lose brain cells? Well, there's a very special place. It's called the hippocampus, and the blue arrow is pointing at it. It's colored in green. If you were to drive a little arrow through here in my head and here and down the top, you'd find that central place. This is our central switchboard of thought and memory. Now, all animals have a hippocampus. Its job is to map the world. Mice and animals use it to find their way around. A mouse uses it to find its den, go out and look for food and find its way back. We use it in much the same way to drive or to walk around your house. As you walk around your house or drive home, you kind of do it on autopilot, don't you? Of course, you may be careful, but as you walk around your house, you know where everything is. You turn automatically around the chair and so on. And in fact, when you stumble into something, it's because someone left the chair out and you didn't expect it. So that's why the little car is there. You can think of the hippocampus as the active memory in a computer. It's a little picture of a computer there. Your thoughts come in through the keyboard, through your eyes. They go to your hippocampus, the active memory portion, and you write them to the hard disk. And when you remember something, you'll go back to the disk and bring it to active memory. So the hippocampus is a pivotal point. While you're listening to me now, or while you read a book, or while you play a musical instrument, you're actually using your hippocampus and actually modifying it. If the hippocampus starts to fail, you have trouble forming new memories. You have trouble remembering things. So it's the link between what's happening and what happened before. Now, let's look at the whole brain. Let's go beyond the hippocampus a bit. You probably understand this image already. This is a side view of a brain, excuse me, and the parts that are lit up are the parts that are active, and the parts that are dark are not very active at this moment. And we transfer information among many brain cells to think, to learn, and to remember. The image on the upper portion of the panel shows the area of your brain that's very active when you read words. Beside it is the area that's active when you hear words. Look, that's a different area. Thinking about words is yet another area, and finally speaking words is yet another area. I tell my students in class that you can sit in a room and listen to me very intently, but that doesn't mean you'll do well on a test, because taking the test is a different portion of your brain. Have you ever heard use a word 8 times and it's yours? Well, what that's actually doing is you're moving it around in your head. So whether you've read the word or heard the word, you have to think about it. It's best if you take and write some notes. It's best if you form your own questions and thoughts. And that really moves the information around in your head. And this also underscores that memories and thoughts and thinking involves your entire brain. It involves so many of your cells in your brain. And if a few are lost over here, you may not be able to move the information around very well. Well, why do we lose brain cells as we age? It's because of oxidative stress. Our whole body is subjected to oxidative stress. But again, my skin is replaced. My liver is replaced. My brain cells are not. So my skin can suffer a little oxidative damage and patch itself up. My brain cells have undergone 61 years of oxidative damage, and they could be suffering for it. What is oxidative stress? You may not know it. You do know what it is. It's rust. Plain and simple. Not so much rust like a little rusty nail as I have in this image, but rust like an apple. Take a bite of an apple or cut an apple in half, set it down on the counter, go away for 20 minutes and come back. What's happened? It turns brown. It's oxidizing. It's literally burning, just like a log in a fire, only it's on a molecular basis. What do you do to make a fire work better? You blow air on it. Oxygen feeds the fire. Well, why didn't the apple burn until you cut it open or bit it? Because it's wrapped in a skin, and the skin of the apple is full of antioxidants. Antioxidants stop oxidative damage. They stop the rust. Lack of antioxidants means our long-lived cells actually accumulate rust. They accumulate burnt, oxidized proteins. How does this happen? Let's dig down into a little bit of the science. We won't go too deep, but this is a special process that happens in all of our cells. It's a cycle, and it's called the methionine cycle. Folate and B12, which you know of as vitamins, they feed into this cycle. There's a little enzyme abbreviated MTHFR. It's methylene tetrahydrofolate reductase. This little enzyme grabs folate and B12 and uses them to make a methionine out of homocysteine. Many people have heard of homocysteine. It's measured in your blood, and it's an index that you haven't been getting enough B vitamins. Because in fact, look at the cycle. Homocysteine is there. If I add some folate and B12, I'll make the homocysteine turn into methionine. Well, methionine is then converted to S-adenosylmethionine, which I'll call SAMe, for S-A-M. SAMe is a miraculous molecule. It's so important to us. Not everyone knows about it. If you think of the most important molecules we need, oxygen and water, SAMe is right up there with them, helping all our cells. SAMe wraps your DNA. It wraps it like a blanket, and it keeps it from breaking down as you age. It takes your antioxidants and makes them bioavailable. You've heard the word bioavailable, haven't you? So you can actually have all the antioxidants you need, but not be able to use them because you didn't consume enough SAMe. SAMe also builds up your neurotransmitters, so it allows your thoughts and memories to move. Well, when you use a SAMe to do any of those jobs, you make a compound called S-adenosylmethionine, which turns into homocysteine. Now, the homocysteine can be exported if there's too much, or we take it and go around the cycle again. So here's where things get a little confusing. Homocysteine is bad for you if there's too much. It's a normal product, and we want to keep using it. Think of cooking in your kitchen. You make some trash. It can't be helped. There's always waste. The problem is if you let the waste pile up. As long as you manage the waste, get it in the garbage, take out the garbage, the kitchen can stay functional and you can make some wonderful food. So as long as we keep this cycle going, we're fine. But if we don't have enough folate and B12, homocysteine is going to pile up. Imagine you can turn the cycle with 10 homocysteines an hour. I'm just picking a number. Well, if you don't have enough folate, you're going to make 20 homocysteines and we can't run the cycle. So what do we have to do? We have to export it. It will go out in our urine through our kidneys. We can use some of it to make an antioxidant we call glutathione, but we can't use the extra to do that. There's just going to be too much. The consequences of too much in our brain are very dangerous, and here's why. Homocysteine interferes with a calcium channel. I'll do this slowly, it's confusing. What's a calcium channel? Well, there's a little picture of that healthy green neuron up in the corner, but now I've got something else. I got these little orange things sticking out of it. I want you to think of a cell as a soap bubble or a water balloon. It has fluid on the inside and fluid on the outside. The outside fluid would be our blood or perhaps our intestinal fluid. The only cells that don't have fluid on both sides are your external skin cells. And of course they're making a layer to hold fluid in us. Well, in the membrane of the cell, if I've got down below a close-up, there are little windows, little channels that open and close and allow us to regulate what can come through the soap bubble or what can go out of the soap bubble. Now this particular slide shows a calcium channel. When the cells need some calcium, including brain cells, there'll be a little signal that says open the channel and you can see some calcium flows in. The signal goes away and the channel closes up. As long as we regulate opening and closing the channel, we can control how much calcium goes into our cells and how much does not, and that's wonderful. And we have channels just like this for sodium, potassium. We have a channel to remove waste, and on and on and on. What homocysteine does is homocysteine acts like a permanent signal, and it hits the edge of that channel. And it's just like go home in the cold and open a window and leave it open. It's way too much. Calcium flows in without being controlled, leading to a whole bunch of oxidative damage in the cell. And everything in the cell can be damaged. If this is a brain cell that you needed for your whole life, we're in serious trouble. And the calcium channels in your brain are very sensitive to homocysteine. So we'll make so much oxidative damage, the membrane itself will be hurt. And more calcium leaks and more oxidative damage in a terrible cycle. Vitamin E, which you know of as a healthy antioxidant, actually protects the membrane. So that's vitamin E's role, and we'll get back to that. But this is the problem that we have with homocysteine, and one of the reasons we have to keep that methionine cycle spinning. Now, oxidative damage disrupts normal cellular events. We refer to these in biology as housekeeping events. Just like you talk about taking care of your own kitchen, the thing your cells have to do to breathe, to make the sugars they need, to dissolve the sugars they need, to make the proteins they need, are called housekeeping. And if you fail at housekeeping, this stuff piles up, and pretty soon you can't cook anymore. So you can no longer make the things you need. The kitchen is simply too clogged. Here's an example of what can happen in vitamin and antioxidant deficiency. What you see in these images is an embryonic brain cell from a mouse, and it's placed in a culture dish and it's growing very well. There's a little arrow pointing to where the axon, the out process of this brain cell, starts. It's a little hard to see the body of the brain cell. It's at the top. Now this is kind of like an X-ray. This isn't an image like you would take with a regular microscope. I'm doing a fluorescent image using a marker that labels a certain protein that lives in the neuron. Think of it as an X-ray. If you took a picture of me, you'd see me like I'm standing here now. If you took an X-ray of me, you wouldn't see all this, you'd see my bones. You'd only see the hard structures. Well, think of your nerve cells, your neurons. You know of an optic nerve, right? You have a retina, and it's got a whole bunch of axons, which collectively we call the optic nerve, that goes back to your brain, it's connected to it. Did you ever wonder how the optic nerve stays connected to your brain? Well, inside the axons are little proteins. They're like Legos. They assemble in a row. And they give a little strength, and they hold up the axon, much like my arm holds up this sleeve. So inside that membrane that forms the axon, it keeps the axon connected to its target so that I can send my neurotransmitters down and connect to the next neuron. There's another very long nerve you know about, the sciatic nerve. It comes out of your spine, goes all the way down to your toes. And again, we need all those little Legos all assembled perfectly. So the little brain cell has to make them and send them out into the axon and assemble them. Well, the little guy that I've been talking about, plus folate, he has his folate. The one next to it, I took away his folate for just 2 hours. And if you look at the arrow in that image, it's a little different, isn't it? I can now see the cell body more clearly. And I see a very big bright spot where the axon begins. What's happening here is this poor brain cell is no longer able to transport the Legos down and get them into the axon. Two things are going to happen. One, it's going to fail to stay connected to the next brain cell, and it'll actually clog up so much like a messy kitchen that I can no longer send neurotransmitters down effectively. This is 2 hours without one vitamin. I'd like you to think of going without enough vitamins in your own brain for a year, for 2 years, for 3 years. So nutrition really does make a difference. Now let me move to something that's a little more complicated, but it's going to show you again, nutrition does make a difference, but more importantly, nutrition can help. That was showing how nutrition or poor nutrition can lead to damage. But I want to examine the combined impact of several risk factors for oxidative damage, and in a result, several risk factors for age-related loss of neurons. Let's start with a genetic risk factor. A weak form of a protein called apolipoprotein E is a genetic risk factor for age-related damage to brain cells. Apolipoprotein E, that's a big word. I'm going to abbreviate it as ApoE. The job of this protein is to clean up waste products of cell membrane damage. So if the cell membranes are rusting, ApoE goes in, cleans it all up, and you can patch up that membrane and keep that cell healthy. Now, that works for all of your cells, but what's the cell that needs to keep its membrane happy the longest? Brain cells that live your whole lifetime. If your skin fails a bit, you can patch it up, you'll make a new skin cell. So it's very critical to keep the membranes of your brain cells healthy and happy, stop the oxidative damage as early as possible. So when ApoE cleans up the waste products, it stops the oxidative damage cycle. ApoE comes in 3 forms. They're called E2, E3, and E4. This is confusing. Let me step away from ApoE for a minute and use an analogy that may help. We all have the same genes. We're humans, but we look different. Why is that? We have different color hair. Some of us are tall, short, some have a big nose, some of us go bald in time. Lighter color hair, darker colored hair, light skin, dark skin. Let's think of eye color. We all have the gene for eye color, but we have different color eyes. That means we have different forms of the eye color gene. Some of us have brown eyes, some of us have blue eyes, some green, some gray. Those are different forms of the eye color gene. And you know what? Blue eyes, although my grandmother used to say how pretty they were, blue eyes are a genetic risk factor. It's a weak form of the eye color gene. Why? Because sunlight can burn my retina. The ultraviolet from sunlight can cause retinal damage. If you have brown eyes, you have a protective form. So I've got a genetic risk. Those of you with brown eyes have a genetic protection. Does that make sense? ApoE works the same way. And if you think about diseases that run in families, heart disease, cancer, and so on, there are often certain forms of genes that work a little better or a little worse. So now, ApoE works just like that. E2 is very strong and protective. It's like brown eyes. E4 is like my blue eyes. It's weak. It doesn't do its cleanup very well. So if you've inherited an E4 form, you are more at risk for age-related oxidative damage and age-related loss of brain cells than individuals that happen to have E2 in them. And you get, what do? You get a copy from mom and a copy from dad, or a form from mom and dad. Just like eye color. If I inherited 2 blue eye forms from mom and dad, I have blue eyes. If I inherit a brown eye form and a blue eye form, I'll have brown eyes. Well, you can have a genetic makeup of ApoE2 and ApoE2, ApoE3 and 2. You get the idea, 3 and 3, 4 and 4, 3 and 4. If you have 1 copy of 4, you are much more at risk for age-related damage. Let's hold on to that. That's a genetic risk factor. And let's consider a nutritional risk factor. This is a complicated slide, but I'll lead you through it. Now, this graph shows 4 conditions. I have some normal mice. That means, in this case, mice that have their ApoE gene working very well. And I have mice that have been genetically engineered such that they no longer have the ApoE gene. It has a minus because the copy from mom and copy from dad are silenced. So normal mice do not have the genetic risk factor. ApoE mice, in this example, have the genetic risk factor. Are we okay with that? Now I'm going to give 2 different diets. A complete diet and a deficient diet. The complete diet has all their vitamins and nutrients. The deficient diet has no folic acid and no vitamin E. The things that move the methionine cycle and the things that help protect the cell membranes. So I take 2 vitamins away, just 2. Everything else is the same. And I feed them these diets for 1 month. Now, they're not injured in any way, they're happy, but now this is like you taking or not taking your vitamins for a year. Mice live about 2.5, 3 years. So 1 month for a mouse is analogous to a year or 2 for us if you extrapolate lifespans. Let's look at what happened in that month. And we're going to measure the amount of rust. Maybe I didn't make it clear. We all form and break rust all the time. The problem is you don't want it to accumulate. So just like a busy kitchen, you're making some trash, can't be helped, but you take that trash out, you don't let it pile up. So the little bar going out across there is measuring how much oxidative damage is present. And this is an average of many mice. So we have a bar and a little hatch line above. That's the range. So we get about 40 mice here that are normal and received a complete diet. And they've got 15 units of oxidative damage or rust. If we take those same mice, or another 40 mice actually, give them a diet lacking folic acid and vitamin E, well, they have just about the same amount of oxidative damage. Everything's doing fine. They're still okay. It's a tiny bit higher, but you know it still falls within the range because look at the little error bar. Look at the range bar above it. So having them go without those 2 vitamins for that month is okay. Again, it's not the best thing in the world for us to do, but they're going to be all right. Mice lacking ApoE, look at them. We gave them the complete diet and they are still pretty close. Statistically, they're identical because they're within that range. Do you see that? But if we took ApoE mice, the genetic risk mice, and we gave them the deficient diet, there's a whopping increase. It's a 25% increase. Not having folic acid and vitamin E for 1 month, and they have substantially more oxidative damage to their brain tissue. So this shows that a genetic risk factor and a nutritional deficiency together make a problem. Neither one alone was serious within that short time. But both of them together was. But you know what? I want to turn this upside down. I told you that the mice lacking ApoE have a genetic risk factor. Yes, they do. But look at what happens. The ApoE mice plus a complete diet are doing just as well as the normal mice. This shows that nutrition can compensate for a genetic risk. Do you see that? Nutrition, in a way, is stronger than genetics. Now, these mice aren't cured. It didn't put their ApoE back in them, but they get along just fine as long as they're taking their vitamins. Now, you're probably aware someone with diabetes, as long as they manage their diet, they're going to be okay. You have a family history of cardiovascular disease, manage your diet and exercise, you can be okay. These data indicate that the same holds true for dementia. You have to work at it, it's not a cure, but your nutrition can help. It can push that genetic risk factor down so that if you do your best, you may not be functionally any more at risk than the person next to you who has an ApoE2, who has the protective form. And there's a little more good news. We lose our brain cells. Remember that little sloping thing I had with the green brain cell and the yellow one? Our brain cells are lost sequentially, not all at once, as we age. Even in Alzheimer's disease, they're lost sequentially. Once you lose a brain cell, you can't get it back. But what I'm trying to say is it's never too late. At any point in the game, if you get in and make a functional intervention, it's good for you. It's obviously better to do it before you've lost any, but it's never too late to start. Now, if you've been listening to the news, you see disappointing studies all the time. A study will come out saying folate supplementation didn't help, or some people took B vitamins and it didn't help. Other people took vitamin E, we see no benefit. There's a new study supporting vitamin E. Why is there one study saying no, one yes? Well, you know what it is? We're very different. We're not all identical mice receiving the same diet. We all have our different genetic makeup. And one nutrient, the change of one nutrient, is unlikely for all of us to show a difference. A single supplement may not make any visible difference. It's not bad. I mean, it's certainly good for you. It's like changing the oil in your car. You won't see a benefit, but long term it's good. So supplementation can be difficult to read out as beneficial, simply because we're very complex. It's a complex regimen that we need. We need exercise. We need a lot of things, all of which I'll discuss. Well, over the years, here at the University of Massachusetts Lowell, my research team developed a nutraceutical formulation from all our different studies, and I only showed you a couple of them, but we studied all of these ingredients. And we came up with a 6-component formulation that makes that methionine cycle spin, that protects brain cell membranes, that provides the energy they need to optimize their function. It balances neurotransmitters. Let's go through the ingredients. The top 3, folic acid, B12, and vitamin E. You know these. These are antioxidants, so we can't make them. They protect our brain cells. They protect all of our cells, but again, 61 years old, want to last another 61 years, not replaced, I've got to take very good care of them. So, and they earn the word vitamin because we can't make them. That's what the word vitamin means, something we cannot make. The other 3 ingredients are nutraceuticals. We can make them, but we can't possibly make enough. So we need to eat them. And since our diet is often deficient, supplementation is in order. The first one, N-acetylcysteine. Whoa, big scary sounding chemical name. Hey, it's not a chemical, it's not a drug. Well, I said that wrong. We are made of chemicals. It's not a drug, it's a natural compound. It's a food metabolite. Look back up at vitamin E. The chemical name of vitamin E is alpha-tocopherol. It's a little scarier than vitamin E, isn't it? So N-acetylcysteine is a building block for an antioxidant that we can make. That antioxidant's name is glutathione. You may have heard of that. It's hard to eat glutathione. Your stomach acid will chew it up. But you can eat N-acetylcysteine and therefore be able to build more glutathione. The next one, acetylcarnitine, is a great energy source. It provides energy for our brain cells. Of course, it provides energy for all of our cells, but when you're not exercising, your brain is taking the lion's share of energy. And finally, S-adenosylmethionine, or SAMe, we talked about it before. It mobilizes antioxidants. It builds neurotransmitters. Now, all of these have been used individually. You can get them all individually at a health food store. They're certainly good for you. One by one, they show some benefit. It's the combination that is effective. And why is the combination effective? Let me show you that methionine cycle again. But I've annotated it now. We add in some folate and B12. So that's going to make more folate and B12 and start that cycle spinning better. We added in some SAMe, so we'll increase methylation of DNA. We'll increase all the things SAMe does. We'll increase neurotransmitter availability. When we use SAMe, remember we make an S-adenosylhomocysteine. Well, adding N-acetylcysteine, I've got it down the bottom as NAC, N-acetylcysteine. That plus S-adenosylhomocysteine will spin down to build glutathione. Well, in doing so, we prevent the formation of excess homocysteine. And hang on a minute, when I'm adding in folate and B12, I grab a homocysteine and make a methionine. So I'm preventing the accumulation of homocysteine, and I'm doing a bunch of other good things. And down in the corner, I've got a little box. Vitamin E and acetylcarnitine don't feed right into this cycle. Vitamin E is busy protecting brain cell membranes, and acetylcarnitine provides energy. As your brain cells are fighting, they need all the energy they can get. So this is how our formulation was developed. There are many other brain formulations that provide antioxidant effect. There are others that just provide the energy. There are others that provide just glutathione, but we're targeting a comprehensive approach. Now, while we're on the subject, why a pill? Why am I talking about supplementation? Why don't we just eat right? Well, I submit to you, we can't. I want you to think back in time to when we were new, cave people, early hominids. We lived as simple tribes, and we lived and looked a lot like these cousins of ours. Has anyone been to the zoo and seen orangutans or the chimpanzees get food? They eat a whole bunch of leaves, and they don't have very good table manners, but we should be eating as many of them, as many leaves and nuts and fruit as they do, perhaps with better table manners. And one of the other aspects is they get their food immediately from the forest and eat it as soon as it's been picked. So it hasn't had any time to sit on the shelf. This is a little bit like the Mediterranean diet. They rarely eat meat. It's not that we can't eat meat. I'll get back into that. But we need a diet rich in very fresh fruit, fresh vegetables, nuts and seeds. We can skip the insects, but the point is that diet is very rich in antioxidants. It's very rich in very fresh green stuff. Broccoli, spinach is rich in SAMe, and we don't get enough. We should be technically munching nuts and leaves all day long, a little at a time, constantly saturating our system with that. So even if you do sit down, you eat some good vegetables every day, it's tough the way we live to eat right. So supplementation is in order. What we've come up with is a perfect storm for age-related cognitive decline. Three things coming together on that poor little hippocampus. We live longer, therefore more age-related loss of brain cells, and the truth is, as we age, our nutrition tends to plummet. We don't pay as much attention as we should to keeping our diet solid. So what I'd like to show you now is one more slide of our science, and then the results of our clinical studies. We took this formulation and went into the clinic. We've conducted 7 clinical trials. Now, 35 publications in our laboratory over the last 10 years is how we built this formulation. We then conducted 5 small clinical studies funded by the University of Massachusetts Lowell itself, with a total of 175 participants. These are published and they're available through the web. They're published in scientific journals from 2008 to 2010. Having completed those studies, we approached the Alzheimer's Association, who granted us funding to conduct our phase 2 studies, 2 additional studies, which we completed in 2012 with another 150 participants. These have been presented at 4 international conferences, including a conference at the Alzheimer's Association last summer in July. They've been submitted for publication. They're under editorial consideration, and they'll be published sometime early this year. We have had no serious adverse events, with a total of over 350 participants, going all the way back to 2006 when we began our studies. What's a serious adverse event? Well, when someone enrolls in a clinical study, there's a very serious documentation, the person's in it, and we monitor how they do, whether they do well, whether they do not, any complications, anything wrong. Should a person move away and have to withdraw from the study? Well, that's an adverse event for the study, isn't it? Should a person fall and break their leg, they have to withdraw from the study. That's an adverse event. They have nothing to do with the formulation. They have nothing to do with what's being investigated. A serious adverse event is a funny term. That means someone had an upset stomach from taking the formulation, or someone couldn't eat, or they couldn't sleep, they were too nervous. None of that. Why? Why none? Because there are no drugs involved. It's food metabolites. And finally, what I'd like to stress is there were no corporate funds involved in any of these studies. These studies have been funded entirely by the University of Massachusetts Lowell and the Alzheimer's Association. Then our studies finished, and my university licensed this formulation to Cevo Nutraceuticals. I'm proud to serve as their science officer, but they did not support these studies. There's no corporate funding behind them. So there's a complete separation between the work we did in the laboratory and the clinic and the now existing product. Here is one of our first studies published back in 2010, with a group of 93 individuals, ages 18 up into their 80s. And we've shown improved cognitive performance for adults. These adults had no known nor suspected clinical cognitive difficulties. Nobody had dementia, nobody had mild cognitive impairment, nobody had any serious problems. Oh, we all forget a thing here and there, but you know what I mean. These are healthy adults in terms of cognition. And every participant had a family member, a caregiver paying attention, and they agreed. So what we've got is a 12-month examination where we dispensed our formulation, the nutraceutical formulation I showed you. It's abbreviated as NF on the bottom of this slide, and it's called the treatment in the graph itself. Or they received a placebo. Couldn't tell them apart. Now this study is double-blind. You may or not be familiar with what a double-blind study is, so please let me explain. If I took my formulation and a placebo, and I gave it to you, and I let you know which one you had, then I look at how you do a month later, 2 months later. Well, that's good, but you know what? I know who got the pills and I know who got the placebo. So there's an inherent bias in there. And of course, I want my formulation to work, so we got to be more careful. So we do a thing called double blind. How do I do this? Well, I take a bottle of the formulation and I take a bottle of placebo and I cover up the label. Or I don't even have a label on it. I have the number 1 or 2. And I know that 1 is the placebo. You don't know it. And I know that 2 is the other one. And I give them to you. And I look away. I leave the room. And you label them A and B. And I don't know which got the A and which got the B. So 1 could be A or 2 could be A and the other is B. So now you don't know which is which. And I don't know which is which. And you dispense them with helpers to all the people that want to participate. So a person participating has a number of their own. I'm participant A1, and I received bottle B. And I'm participant A2, and I received bottle A, and so on for our 93 participants. So nobody knew what they had, and no one who gave it out knew what they had, and I didn't know what they had, and pretty much no one knew anything. But we know we have 2 groups, the A group and the B group. So we looked at everybody's performance on a special test at the baseline. Then we gave them those formulations, A or B, and then they went away and they came back 3 months later, and we measured their performance again. We used a special cognitive test here called the trail making test, and I won't go into very much detail, but it's simply a timed test. How well do you do it and how fast do you do it? Your job is to connect the dots on a piece of paper, and the dots are numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and lettered A to L. And your job is to connect them in sequence as follows: 1A, 2B, 3C, 4D. And they're not like that. They're all spread out. So 1A, 2, 2B, 3C. And that's easy. But when you get up to G, what number? So F, 6, G, and it takes about a minute to do. So you know what? If you're thinking a little faster and sharper, you'll do that a little better. Well, there's the baseline. We normalized everyone's baseline to 0. And we looked at how they did after 3 months. People receiving the treatment improved. And they are statistically different from those receiving the placebo. But you know what? Who wants to be on a placebo for 12 months? So what we arranged with everyone is, please, you're going to receive pill or placebo, nobody knows it, for 3 months. But after 3 months, I promise you, we're going to break the code. You're going to see what you had. You'll see your own score. But then you're all going to receive the formulation for the rest of the time. So at 3 months, you can see the little squares on the bottom. The placebo group wasn't doing as well as the treatment group, but at that point, we broke the code and everyone received the formulation. So the placebo group is called a delayed start group. They started 3 months after the people receiving the formulation. See that? And look at their performance between 3 months and 6 months. The group that used to have the placebo, that delayed start group, caught right up to where the first group was at 3 months. And the guys receiving, guys are he's and she's, the guys receiving the formulation from the beginning, they still improved a little bit. Now at 6 months, we did something very special. We took the pills away from everyone. Nobody received any more pills for 3 months. And look what happens. They decline in performance back to where they used to be. This proves they didn't memorize the test. And it also confirms you need to keep taking a supplement to reap its effect. And you know that. You're on the treadmill every day, you're losing weight. You get off the treadmill, stop using it, what happens? Probably going to gain that weight back again. So you have to stay with the program. Well, at 9 months, we gave the pills back to everybody, and indeed, they improved again. So this is wonderful improvement, a double-blind test over the course of 12 months. We were astounded at how well this worked. We never expected it to be this great. We're hoping to preserve function. We actually see improvement. We got very excited and said, well, that took 3 months. Does it take 3 months, or what's happening in between? So we did this test again with 43 different individuals. Again, with no known or suspected cognitive problems. And we gave them the formulation under that same kind of double-blind setup. And we examined how they did after 2 weeks and 4 weeks. And look, there's statistical improvement within 2 weeks. And further improvement by 4 weeks. And when we took that placebo group, switched them over to the formulation, they caught right up. So one can expect, on average, improvement within just 1 month. Everyone's different. There's a range. Some people improved a lot. Some people improved a little. Some people held their ground just well. Some people, tremendous improvement. It averages out to the line you see here. But again, within 2 weeks or just 4 weeks. We also worked with a bunch of individuals with diagnosed cognitive decline. Now, different tests may be appropriate in that case, and I'm showing you one of them here. This is the work that was funded by the Alzheimer's Association, part of our phase 2 study that's now under consideration for publication. And we used, in this case, the clock drawing test. Now, I show you a couple of images of a clock here. This was drawn by an individual diagnosed with Alzheimer's disease. He's 88 years of age. He had been on one of the pharmacological treatments, but he had complications and had to stop. So he started taking our formulation. Now, the clock drawing test is a simple test. It is standard, as is the trail making test. So if you went to a neurologist or a doctor who's concerned about your cognitive performance, they may ask you to do this test. And it's done as follows. You sit in a room and relax. There's no clock in the room. You're handed a piece of paper, a pen, and you're asked the following. Are you ready? Please draw me a clock, the face of a clock. Please put the numbers in it. Please space the numbers out nice and evenly. Please put the hands of the clock on it. Please make a big and a little hand so a child would know the hour and minute. And please put arrows on the end of the hands. And please make the time say 12:45. It's easy, isn't it? No, it's not. It's incredibly difficult. And we forget. This is the power of humans. This is how powerful our brain is. I can give you a list of words and say those words back to me. That is memory. Sure it is. But, you know, it's a simple kind of memory, isn't it? A parrot can learn words. Your dog or cat knows certain sounds mean get off the table or get away from that or come on, I'm going to feed you, and so on. Right? Something like this is testing our executive function. What do I mean? Well, you have to execute. You have to picture the clock in your mind, visualize it, and execute it in a series of steps. This is analogous to remembering a cooking recipe or a complex list of tasks. And someone with dementia, depending how severe, is essentially going to lose their place partway through it. Right? And you can see that this poor individual had a lot of problems in the beginning. Let's just think of the time. 12:45, if you get all the way to the end, 12:45. That's tough. An elderly person might say 1:45. But even a person who's familiar with a digital clock, 12:45, there's no 45 on the clock. You have to think through, where is 45? Where is quarter of? And so there's no 4-5 to point at. You have to realize that means go over here. And if it's 12:45, the hour hand doesn't point at the 12, it doesn't point at the 1, it points in between. Does that make sense? You gain a point for each one of these things you get right. Well, this individual took our formulation for 9 months. Look at his performance. He's not perfect, but he's much, much better. What we've probably done for this guy, we didn't look inside, but his neurotransmitters are kicking again. He's able to form thoughts a little better. The graph that accompanies this image is a graph of 112 people who received our formulation or a placebo for 3 months, again, double-blind, not knowing what they received. So we normalized all the starting scores to 0 so everyone can be on the same scale. The people receiving nutraceutical formulation, NF, you can see they've increased. 68 people increased from baseline statistically. People receiving placebo, 44 of them didn't get off the baseline. Now, we followed these people for 9 months. There is long-term improvement for individuals with age-related cognitive decline. This includes many people that were diagnosed with Alzheimer's disease. The placebo group is presented in red. The people receiving our nutraceutical formulation are in blue. And we get that same 3-month split that you just saw in the last graph, but now I'm carrying it out for 9 months to show you. Well, at 3 months, we did the same thing we did in that earlier study with healthy adults. We switched people from placebo onto the formulation. So they don't really have improvement over those first 3 months. There's a little bit of an uptick because they're involved in a study, they're feeling good, but it's not statistically improved. Once we switch them to the formulation, you can see they catch right up to the rest of the individuals on the formulation, and at 9 months they're doing much, much better than they were doing in the beginning. Now, this is wonderful. This is great improvement in cognitive performance and executive function. We're very happy that people receiving this formulation also have an improvement in mood. Those of you that know someone with dementia, with Alzheimer's disease, either a family member or a friend, you may be aware that mood, the person can become very irritable, very obstinate, angry, depressed. And this is really because things are getting confusing. So it's related to cognition, but it's kind of another facet, isn't it? And mood can be more difficult to handle than actual memory. If you've got a spouse or a parent or a sibling or anyone you care for, and you have to answer the same question 10 times, what day is it, what day, that's annoying. But if that poor person is yelling, lost their appetite, if they can't dress themselves, they can't use the bathroom, they can't use the phone, they wander, how do you go to the store? Never mind hold a job. Those are the issues that can tear the family apart. And we're very happy to see that our formulation also had a mood benefit. Now, this is a caregiver report. If I were the participant, you'd ask my wife, how is Tom's mood? Is he depressed? And she'd say yes, no, and if yes, she'd grade my depression as 1 to 5, 5 being more severe. Is he irritated? Is he angry? Is he agitated all the time, nervous? She'd say no or yes, and if yes, score it 1 to 5. Has he lost his appetite? And on and on and on. There are many criteria. So the caregiver of those individuals you just saw the cognitive performance for, the caregivers completed this test. It's called the Neuropsychiatric Inventory, and it's also a standard test. Now, a perfect score is a 0. Everything's fine. I'm not agitated, I'm not irritated. We'll probably get a 1 or 2. We're not going to get a perfect score. But you can see these individuals are already at 11. They've declined. I get that y-axis upside down because decline, more numbers are declining, not getting better. So it's easy to visualize this way. So our participant pool that you just saw the cognitive performance for, they're already down to 11 on this scale. And the people receiving placebo, they keep right on declining over those first 3 months. But those receiving the formulation, they stop declining. They got a nice plateau going over 9 months. Well, remember, we switched the placebo people onto formulation after 3 months, and when we do so, they get a little bounce back, and they're holding their ground at 9 months. So we have a cessation of the mood and behavior progression, a cessation of that decline, and that's a wonderful, wonderful benefit. Now, those are some of the clinical data, and the studies that we've published are available. I'll show you a website in a bit, but let's step back for a minute. I've shown you some great results with this formulation. Is it all you need? Absolutely not. We're very happy to see these results, but let's back up. Let's go to the basics. You need a diet rich in fish, green vegetables, fruits, nuts. This is the Mediterranean diet. Have you heard of that? It's the antithesis of the Western diet, which is steak and potatoes and fried stuff every night. Now, the Mediterranean diet does not mean you have to get your food from Europe, which someone asked me once. You need fresh fish, green vegetables, right from your supermarket. Go to the best supermarket you can, get the freshest stuff you can. Go to the best health food store and get some fish in your diet. Get some very fresh vegetables in your diet. There are other supplements that help. Remember, supplements in isolation, little or no impact. Put them together. There are many other brain supplements that you can see. Well, they give you antioxidant protection. They're not bad for you, but I submit to you they're not going to target that methionine cycle and do some of the things we've shown you. If you listen to nothing else I've told you today, take a good multivitamin. You hear of problems every so often from multivitamins. That results from overdosing, not from the correct levels. Talk to your pharmacist, your doctor, go down to the closest health food store you can and ask about the best multivitamin. I have no recommendation to make, but I do recommend get a good one and take it. So now we're dealing with the basics, a Mediterranean diet and a good multivitamin. And then you could put my supplementation on the top of it. That's wonderful, right? But don't try to add a supplement and keep those things lousy. It's almost like pouring something good on a bad foundation, right? You can have red meat. You certainly can, but just not all the time. Everything in moderation. Some people say, oh, I hate the smell of fish. You can get fresh fish that doesn't have an odor. Some people say, I hate the taste of fish. Then get an omega-3 pill. Go down to your health food store and talk about it. There are supplements that will help. Now, I'm very happy to say our nutraceutical formulation is now available as a product called PERCEPTIV, marketed by Savo Nutraceuticals. This is the same formulation used in our clinical studies. It's actually made by the same manufacturers. My university, UMass Lowell, licensed this formulation to Savo Nutraceuticals, and I serve now as their science advisor. So when there's a question of someone, I'm there to help out. The components of this formulation are made in the United States in a certified CGMP facility. That stands for Current Good Manufacturing Practices. Each bottle has a 30-day supply. You take 2 tablets a day. I know from our clinical studies you can take the 2 tablets in the morning, or you can do 1 in the morning, 1 in the evening. Either way works. Do what you remember. Do what you're not going to forget to do. It sounds like a bad joke. I don't mean it that way. I take my 2 in the morning along with my multivitamin and other supplements. Please, here's the website. For more information, visit www.thinkperceptive, there's no E on the end, thinkperceptive.com. You can download my clinical studies from that site. You can learn more about it. You can learn where it's available at a location near you. We had all kinds of testimony from individuals during our clinical studies. These are things that they just don't go on a graph. They're things that just make you feel wonderful. Someone said that their grandmother remembers and sets the table for dinner now. She didn't for the longest time. Her grandfather was talking and playing with his grandchildren again. The company continues to receive such testimony. Someone said I was more alert, in a better mood. I noticed increased energy. Weeks later, I'm starting to notice changes, a sharper memory, a better mood, and so on. So please, in closing, please remember, any supplement, this supplement, any supplement, is one part of a healthy approach to aging, memory loss, and cognitive decline, whether or not you ultimately face Alzheimer's disease. Importantly, supplements do not replace a drug prescribed by your doctor. Supplements, nutrition, the Mediterranean diet is good, but please don't delude yourself. If your doctor has recommended a prescription and you say, I'm doing all these natural healthy things, I don't need that drug anymore, ooh, be careful. It's very dangerous. Do not self-medicate, and similarly, do not stop self-medication. You want to do a strong dietary program and you have issues and you want to know, do you still need the prescription? Talk to your doctor. Bring in all the things you're doing and discuss it. Maybe there'll be a modification, maybe not. But don't make that decision yourself. Now, supplementation, no supplementation, you need a good balanced diet. You still must exercise your brain. Please, do a crossword puzzle. Do that Sudoku. If you watch television, try to watch active television at least part of the time. Watch the National Geographic Channel, the Animal Planet. Watch Through the Wormhole. There was someone sitting in an audience once, and his spouse kind of elbowed him. And I said, what's the matter? She said, well, he watches sports all day long. Well, you know what? If you watch sports all day, well, not all day, but if you're watching sports, be the coach. Think of the next play. Think of what they did wrong. What would you have done? What could you do? What could that player have done? And you make your brain active. It all doesn't have to be book learning. The point is to think, not be passive all the time. Does that make sense? There are computer games that help. The games we had when I was younger, Pac-Man and those little things, they don't really help you. But if it's a game where you remember where something is and you have to turn the card over and such, that actually stimulates your brain. That's a good thing. Nutrition, supplements, healthy lifestyle, they're not a cure. They're a proactive approach. There is no cure. We're biological creatures. There is no cure for age-related cognitive decline. But you can be proactive and make a difference. So thank you very much for your attention. Be well.